Hello, and welcome back to Mechanochemistry Discussions. This seminar series is hosted by NSF-funded Center for Mechanochemical Controlled Chemistry, or CMCC. My name is Mateusz Mariański. I'm one of the center's investigators. As always, one of the goals of this seminar series is to bring our community together. The seminar streams live on the third Thursday of every month at 11 a.m. Eastern, and they are also available to watch anytime on the YouTube channel. Also on YouTube, you will find all previous speakers that participated in the series, and I encourage you to check them out. We also have some excellent speakers planned for the upcoming months, and we hope that you will join us for all of them. Before we get started, a few quick thank yous. First, Dr. James Batias, the Center's Director, Jennifer Belcik, the Center's Administrative Coordinator, Francesca Antillon, our Outreach Coordinator, and a CMCC student without whom the seminar would not happen, Ryan Klok. Please do continue to follow us on our YouTube channel at CMCC Mechanochemistry Discussions. Before we start, a few quick guidelines. This seminar is being recorded. If you have any questions during the webinar, you can either email them to us at cmccdiscussions@gmail.com or you can post them directly into the YouTube channel. Either way, they will be propagated to the speaker at the end of the seminar. We also created a short survey to learn more about our viewers, what you think of this series, and how we can make it better. If you want to provide some feedback, please scan the QR code or click the link at the end of the description of this webinar on YouTube. Without further ado, it is my pleasure to welcome today's speaker, Professor Harm-Anton Klok from EPFL. Harm-Anton Klok is a full professor at the Institutes of Materials and Chemical Sciences and Engineering at the École Polytechnique Fédérale de Lausanne. He studied chemical technology at the University of Twente from 1989 to 1983 and received his PhD in 1997 from the University of Ulm after working with Martin Müller. After postdoctoral research with David N. Reinhold, again at the University of Twente, and Samuel I. Stapp at the University of Illinois at Urbana-Champaign, he joined the Max Planck Institute for Polymer Research in Mainz, Germany in early 1999 as a project leader in the group of Klaus Mühlen. In November 2002, he was appointed to the faculty of EPFL, and from 2012 until early 2025, he served as a director of the Institute of Materials at EPFL. It is my pleasure to welcome Professor Klok to Mechanochemistry Discussions, and the title of his presentation will be Mechanochemistry of Solvent-Swollen Surface-Grafted Polymers Brushes and Polymer Networks.
The audio is good?
You can begin.
Okay. Hello. Thanks a lot for the opportunity to share some work from our laboratory in this online seminar about the effects of solvent swelling on the mechanochemistry of polymer brushes and polymer networks. I'd like to start this presentation with a brief intro. First of all, with acknowledging the people who have done all the work. The story I'll share today is actually a summary of up to 2 decades of research of a number of students and postdocs that have worked in the laboratory since 2003. And it also shows a bit how the projects have conceptually evolved over time. So I'd like to start with a few words of introduction to set the scene and say a few words about the history of polymer mechanochemistry. Polymer mechanochemistry is a subfield of polymer science that is interested in understanding and also utilizing the effects of mechanical force on polymers. And one of the first experimental observations that illustrated this effect goes back to the 1930s in a paper published by Hermann Staudinger, in which Staudinger and a co-worker reported that mastication of polystyrene was found to result in a gradual decrease in the viscosity of polymer solutions and a corresponding decrease in molecular weight of the material as a function of the mastication time. And Staudinger in 1934 already, this is an excerpt of the paper, already proposed that these observations were due to the scission of covalent carbon-carbon bonds in the polymer main chain. This observation is quite interesting actually, because Staudinger, as you may know, was the one who actually proposed the existence of macromolecules, and this paper on the observation of mechanochemical scission of macromolecules was actually published at a time where this concept of macromolecules was still under debate. Staudinger, as a matter of fact, only got a Nobel Prize for conceptualizing this idea of macromolecules in the 1950s. So the first observations that described the impact of mechanical forces on degradation of macromolecules go back to the 1930s. And these observations then form the starting point of this field of mechanochemistry. In the decades afterwards, from all the way up to the end of the 20th century, a lot of researchers spent quite some time to fundamentally understand the effects of various modes of force activation on polymer degradation. This includes, for example, ultrasonication studies, the effects of high flow shear fields on scission of macromolecules and on molecular degradation. All of this primarily for the context to understand changes or impact on molecular weight, as this is important in determining the properties of synthetic macromolecules. Then around the change of the century, there was kind of a conceptual advance or change in the field where people started to realize that it may be interesting to design macromolecules that incorporate certain functional moieties, highlighted as this ellipse word in the slide over here, which were referred to as mechanophore, and which were designed to be molecular units that are strategically weakened from a mechanical perspective as compared to the rest of the synthetic macromolecule. And the idea was then that force activation of these macromolecules could then be utilized, for example, to generate radicals, to trigger color chains, to release monomers or other active moieties from the polymer scaffold. The idea being that rather than just considering force as a destructive effect on a macromolecule, try to think about harnessing forces in a positive way and to endow macromolecules with new materials properties. And another interesting thought to add to this is that typically the field of mechanochemistry has focused on investigating the effects of fairly high forces on macromolecules, typically a nanonewton and higher, where a nanonewton is typically defined as ballpark a force that one would need to break a covalent bond. And it's interesting to compare this with nature, with biology. Biology also utilizes or makes widespread use of forces to modulate properties of biological matter, but nature does so utilizing forces that are significantly weaker. Examples include, let's say, protein unfolding processes or rupturing protein metal ligand bonds that operate at lower force length scales. So one observation here is that the polymer mechanochemistry community typically looks at rather high forces, but it may be interesting to take inspiration by biology and also think about maybe the lower end spectrum of this graph and try to understand how and whether that could affect polymer properties. An interesting question is basically, if one looks back at the history of the field of polymer mechanochemistry, typical processes that have been utilized to study the effect of force on polymers include ultrasonication, high flow shear fields, but also more traditional things as basically pressing, hot pressing samples or tensile testing. And almost a decade ago, there was a paper published by this researcher in Strasbourg that kind of proposed the idea or this concept of soft mechanochemistry, in which they asked the question how polymer materials would potentially react to lower energy demanding processes. And they gave a number of examples to this, also largely inspired by biological processes, some of which I referred to on the previous slide. And so if you look in the polymer literature, you can find actually quite a large number of examples of processes which are activated by putatively low energy processes, in particular solvent swelling, and that can trigger reactions that involve the cleavage of covalent bonds. There are papers that have reported this on basically in solution-based systems, for example, bottle brush polymers, but also 2D systems, polymer brushes, as well as 3D macroscopic systems. And I'll give a few examples of those. And this is a part of the field where we are quite interested in, and where I would like to show some examples about. And there is one example that I find quite interesting, and that drives quite some of the questions we're looking in the lab. This also comes from a paper that was published a couple of years ago already. The experiment that's illustrated here utilizes a cylindrical type gel specimen on the upper left corner. It's a cross-linked polyacrylic acid hydrogel. The cylindrical specimen is a piece of hydrogel where the upper part and lower part have different cross-link densities. The upper part has a high cross-link density, the lower part has a lower cross-link density. If you put this gel into a good solvent, the cylindrical gel will swell, but obviously the lower cross-linked bottom parts will swell to a higher degree than the upper part. And what the authors observed when they swelled this gel, as you can see in the picture on the lower bottom, is that basically exposure to a good solvent basically results in rupture at the interface between the lower and higher cross-linked gel. And so one question we are kind of interested in is try to understand basically what is the molecular origin of basically these type of macroscopic failure processes in soft materials, keeping in mind that what we see here involves breaking covalent bonds and is essentially only driven by solvent uptake of a covalently crossing network. And so in the lab, we are interested in trying to use two sorts of model systems to obtain insight into these questions. One, basically a 2D system and a 3D bulk system. The 2D systems we're interested in are chain end tethered polymer assemblies, which are referred to as polymer brushes. And in parallel to this, we're also interested in basically studying similar types of effects in three-dimensional cross-linked polymer networks. And for the sake of time today, I will primarily focus and illustrate some of the ideas we're trying to pursue by giving some examples of our work on chain end grafted polymer brushes. Also, because that's a little bit of how we came to work on these types of problems. So the way I like to see this research is basically we're interested in utilizing the surface-grafted polymer brushes essentially as some sort of molecular tensile testing machines. A surface-grafted brush is basically a bunch of chain end grafted polymers tethered to a substrate. And I'm interested in trying to see if we can modulate tension on these surface-grafted polymers by playing around with solvent quality and solvent uptake and the swelling. And the idea is if we can use that to modulate tension at the interfaces, and then if that's a way to trigger reactivity of surface-grafted polymers. So we're interested in a few questions. Question one is basically whether swelling of polymer brushes accelerates chemical reactions. Can we control rates of these reactions by controlling degrees of swelling being proportional to force? And if we can figure out ways to actually measure these processes, so can we capture this somehow and describe in a quantitative or semi-quantitative fashion? So as I mentioned in the beginning already, the story of this presentation dates back literally 2 decades. So I would like to start with basically some historical work to illustrate how we came to work on this. So the history of the story is that about 2 decades ago, we were very keen on grafting polyethylene glycol methacrylate polymers from glass and silicon surfaces with the idea that we want to generate biologically inert surfaces and use them as substrates for cell culture studies. So these polyethylene glycol methacrylate brush films, I'll show on the next slide, were grown via surface-initiated atom transfer radical polymerizations from glass substrates. And we observed if we incubated these films grafted from glass slides into cell culture medium, that there was delamination which could be observed on these substrates. And kind of we were asking the question, and we want to understand basically what drives this and what's the reason for this delamination. So our working hypothesis to describe this delamination process is schematically illustrated in cartoon on the upper part of the slide. So on the left part, you can see a very simple cartoon that illustrates this polymer brush. The monomer we utilize is hydroxyatom methacrylate. These brushes are grafted from silicon surfaces using organosilane anchored initiators, where an ATLP initiator is tethered via an amide bond, and then we grow these methacrylate brushes from the bottom up. You should consider this brush on the left hand somehow as a brush in a dry state. If we place this brush into a good solvent, an aqueous medium, the solvent will swell the polymers. These polymers will undergo chain stretching, which means that the thickness of this brush film in the middle is higher than on the left hand. Typically, swelling results in a 2 to 3-fold increase of film thickness. The other thing that you need to keep in mind is that if you conduct surface-initiated atom transfer radical polymerization of a monomer such as hydroxyatom methacrylate, strictly speaking, we are not generating a true brush in the sense of basically a bunch of polymer chains that are tethered to a surface. But ATRP of hydroxyatom methacrylate results in a very small fraction of interchain crosslinks, as we have illustrated here. And this is due to oxidative side reactions and due to trans-esterification reactions that accompany the surface grafting process. So strictly speaking, what we have here is not a swollen polymer brush, but basically a solvent-swollen thin hydrogel layer. Then the hypothesis we had was it upon swelling, water in the medium cleaves these siloxane bonds that tether the anchors of the polymer chain to the substrate. And if we hydrolyze a sufficiently large number of these anchoring bonds, due to the cross-linked nature of the film, this would basically result in degrafting. And since the film is cross-linked, that's the reason for the delamination that we observed. So this was the hypothesis, but we want to understand this a bit more in depth and investigate it in a bit more systematic manner. And with the experiments here, there are two problems. Problem one is, of course, that these brushes are not true brushes, but they are lightly cross-linked films. So the only thing we can observe is basically whether they delaminate or not. But a continuous monitoring of a degrafting process is a bit problematic. The second part relates to the fact that basically we are studying here hydrophilic brushes in an aqueous medium, and the aqueous medium, water, at the same time is the solvent that basically swells the film, but also provides the reagent for the chemical reaction we are considering here, which is an hydrolysis reaction. So we needed to redesign the system a bit to be able to have a more systematic look into this. So our ideal experiment was one where we have a true brush, so basically just chain and tethered grafted polymers with no interchain crosslinks. If we break or cleave the bonds between the engrafted polymers and the substrate, this will result in degrafting. Degrafting reduces the number of chains per unit surface area. It's called grafting density. And since the grafting density is reduced, these polymer chains will be able to adopt a more collapse conformation, which then basically means that if we monitor the dry film thickness of these systems as a function of time, we actually have a method in hand to observe this degrafting process. This is element one. Then the second question is basically, how do we design the system? So for degrafting, we need a good solvent to swell the brush, and we need a reagent, in this case water, to induce chain cleavage. But if we use a hydrophilic brush, the solvent and the reagent are the same, so we can't really decouple the effects of solvent swelling and of the chemical reaction. So to overcome this, we designed a system where we utilize not a hydrophilic, but a hydrophobic polymer brush. So the brush systems in this slide were generated by atom transfer radical polymerization of T-butyl methacrylate. So that's a hydrophobic and non-water soluble monomer. And we decided to look at swelling and possibly degrafting of these hydrophobic brushes in organic media that would be miscible with water, our reagent, to some extent. So in this way, we're able to decouple the effects of swelling and chemical reaction, because we can deliberately add specific amounts of water to our reaction medium. And as I mentioned before, the experiment basically consists in incubating these polymer brush systems in a good solvent for the brush, let's say THF or water, either in the presence or the absence of a small amount of a reagent, water in this case. I mean, as a function of time, we analyze the evolution in dry film thickness of the sample, which then indicates whether degrafting occurs or not. So if you do this for a poly-T-butyl methacrylate brush film, and you test 4 different media, toluene, THF with 11 ppm water, or toluene with 330 ppm water, or pure water alone, and you measure dry film thickness as a function of time, essentially we don't see any appreciable or significant thickness as a function of time. So the conclusion from this is, in a solvent like toluene, it is a good solvent, the brushes swell, but there is no water, so no reaction. Pure water is a bad solvent for the brush doesn't swell, so there is also no reaction. And these two THF, the THF and the toluene water mixture do contain some water, but the amount of reagent is not sufficient in order to produce chain cleavage. So then we went on and we studied a bit more in depth THF water mixtures. And I'd like to focus your attention first on the data set on the left. So this shows dry thickness as a function of time. This is a period of 7 days. First for a brush that was incubated in dry THF. So you can see dry THF is a good solvent. I'll comment on this in a second. In this medium, the dry film thickness does not change over time. So the system is stable. If you add to this THF 5% water, these are the triangles, you will see a very rapid decrease in dry film thickness to a low baseline number within about a day of time. And if you increase the amount of water from 5 to 20 volume%, you will see a similar decrease in dry thickness, although a bit less. And it's interesting to correlate these observations with the swelling ratios of these films, which are plotted on the right hand. So the swelling ratio of a polymer brush is the ratio of the dry film thickness, of the swollen film thickness, in particular medium, divided by the dry film thickness, which we use as a measure for the extent of chain stretching of these polymer brush samples. And if you look for the brush sample with an initial thickness of 160 nanometer at the left hand, you will see that the swelling ratio in THF is about 3.5. If you add 5% water, the swelling ratio does not appreciably change. It basically means the black squares and the blue triangles in the left data set are data points for a brush where the chains are stretched to a similar extent. In case of the blue experiment, there is water, black, there is no water. Water results in chain cleavage. If you now increase the amount of water from 5 to 20%, you can see in the data on the right hand that this results in a small decrease in the swelling ratio from about 3.5 to about 2.5. And as a consequence, this is consistent also with a somewhat reduced rate of degrafting that we have observed in that medium. So this already indicates two things. It indicates that degrafting occurs indeed if sufficient water is present. So we have to go to 5 volume%. And if we compare the data set on the left with the swelling ratios on the right, there also seems to be a correlation between the rate of degrafting, which we take as the slope of this curve of these data sets, and the swelling ratio. So between the rate of a reaction, quote unquote, and the extent of chain stretching of these surface-grafted polymers. This is also plotted here in a slightly different way. So if we look again at the evolution of film thickness versus time, and if we basically take the initial slope of the decrease of film thickness versus time as the initial rate of degrafting, we can plot this on the right hand as a function of the initial swelling ratio of the polymer brush. This is only a three-point graph, so it doesn't say a lot, but it illustrates basically if you compare THF plus 20% water and THF plus 5% water, that there is a positive correlation between swelling ratio, which is a measure of chain stretching, and the initial rate constant. So this is just one solvent medium with two different amounts of water. But it suggests that the initial rates for degrafting correlate with the initial swelling rates of the brushes. And we want to kind of substantiate this by exploring a number of different organic water-miscible solvents, just to have a few more data points and try to further substantiate this correlation between this apparent rate for this degrafting reaction and the initial swelling rates of these systems. So on the left, you can see again the evolution of dry film thickness versus time in THF plus 5% water. That's a data set I've shown before. And for comparison, also results in acetone and DMF with a similar water content. So we chose those other solvent mixtures simply because they have different solvent qualities for T-butyl methacrylate, and it is a way to modulate the swelling ratio. And the plot on the right of this slide essentially presents this apparent rate constant for the degrafting reaction as a function of the swelling ratio. And this graph basically illustrates or suggests that there is a positive correlation between the swelling ratio of a brush. So the more we swell it and the better the solvent, the higher the apparent rate constant for the degrafting reaction seems to be. There are a few things to note. For the reaction rates for hydrolysis of these organosilane bonds that we think are involved in this process are quite similar across a variety of organic solvents, including those that we have looked at, which makes us believe that basically the effect of solvents is less important as the effect of chain stretching induced by swelling. And the same thing also that these reaction rates for ester and imide hydrolysis, which could also contribute, are known to increase with dielectric content of the solvent, which is actually opposite to what we could observe or extract from these data points, which supports the idea that what we're observing here is correlated to this effect of chain stretching of these polymer grafts. The data on the example on the previous slide was an example where we utilized hydrophobic polymer brushes, and we used organic solvents of different solvent quality to control the degree of swelling, and water as a reagent. We've done a similar set of experiments in aqueous media, where we studied poly-3-sulfol propyl methacrylate brushes that we also prepared for ATRP. And for these samples, we've tried to study the degree of swelling by tuning the amount of salt added to the aqueous media. The experiments, by and large, are very much the same as I've shared with you on the previous slide. And if you look at the graph on the lower right corner, we can see that this apparent rate constant for the degrafting reaction also correlates here with a swelling ratio, which we modulate by the amount of salt added. With the caveat, of course, that the error bars here are quite large, which has to do with the fact that the swelling ratios are obtained from swollen polymer brush thicknesses determined by ellipsometry, which is non-trivial tasks to determine in some cases with a high precision. But the correlation seems to be consistent with what we have observed for these hydrophobic systems a slide earlier. So the conclusion of this work is that basically that it seems to indicate that swelling-induced chain stretching of these end-tethered polymer brushes seems to activate bonds at the brush substrate interface. We believe this is primarily the siloxane bonds, but it could also be the amide and the ester bonds that tether the ATRP initiators. And the experiments that I've shown to you basically suggest that one can modulate this tension by basically solvent quality, which then translates in basically swelling ratio. So as I mentioned before, we are quite interested also in trying to, this is basically an observation. The question now is basically, can we also measure the forces that act at the interface between these surface-grafted polymers and the substrate? And a very interesting way to try to come up with a system to measure those forces, or at least to visualize them, is to try to take advantage of some of these force responsive, some of these color responsive mechanophores that have been developed, which undergo a color change upon force activation. And so our idea was to try to see if we could take advantage of this and design systems where these force probes would be integrated at the interface between the polymer brush and the substrate, and to try to see if these force probes could also be activated by solvent swelling of our polymer brushes. And the idea that we have, or one vision, is that basically if you look at the landscape of force probe that exists, there is a quite broad range of force probes available. Some of them act in the at the higher force ranges and are quite hydrophobic. And there is also force probes available that are inspired by the biochemical community that work in the lower force end. And there may be some room to develop some force probes that basically would help to map out forces in the intermediate range. So these molecular tension force probes, we believe, could be a quite interesting tool to understand basically mechanochemical processes at the brush interface. So we've done some first experiments to try to design molecular tension force probes that could be integrated in surface-grafted polymer brushes. The idea being that potentially one could take advantage of thread-type phenomena to assess, ideally quantitatively, the forces that would act upon basically solvent swelling and stretching of surface-grafted polymers, as illustrated here. And in the slides that follow, I want to give you some preliminary data that demonstrates the synthesis of these surface-grafted thread pairs, and also some preliminary data that give you a first qualitative idea of the effects of solvent swelling of such systems. So the thread pair we opted for was a published system which contains of two dyes, an electrolytic dye and another dye called QS21. These dyes were connected via a well-defined oligoethylene glycol spacer composed of 24 repeat units. This requires multi-step synthesis. This peptide was then endowed with an alkyne group at one chain end, which allowed us to immobilize the systems via a click-type reaction on the surface, resulting in substrates which both present an ATIP initiator, as shown here, that is immobilized in a normal way to the surface, but also in ATIP initiators that are immobilized by this force probe. And then this was used to graft dimethylamino acyl methacrylate brush films from silicon surfaces. And in the first set of experiments, which are shown at the bottom corner here, we tried to observe possible thread effects upon exposure of these surface-grafted films to PBS medium. So the image on the left, panel A, shows a fluorescent micrograph of a dimethylamino acyl methacrylate film that incorporates a thread probe in a dry state. So basically here, fluorescence is quenched, there is no thread. If we take this brush and basically incubate in a good solvent, PBS, the brush swells. Thread is basically, you go to a low thread state, and we see a red fluorescence appearing, which is consisting with basically chain stretching. And then if we incubate the system for about 2 days, which is the time frame in which we know degrafting occurs, we see that basically there is no longer any thread presence, which would be then consistent with degrafting and collapse of the polymer layer. So this is the first basically qualitative proof of concept experiment that shows the incorporation of these thread pairs at the interface of surface-grafted polymer brushes, which we think is maybe an interesting avenue to at least visualize and maybe even quantitatively analyze some of these processes. I think that brings me already to the end of the presentation. So the idea of the presentation was to give you an overview or an appreciation of the effects of processes which are putatively simple, so simple solvent swelling processes, and that these processes can have quite dramatic effects on certain types of polymer materials. I've used surface-grafted polymer brushes as an example, where under particular conditions, solvent swelling can lead to degrafting. So it's an important process also from a practical concept, if you think about stability of surface-grafted polymer layers. I believe that the experiments are quite illustrative in the sense that they suggest that there is a direct correlation between the extent of chain stretching as controlled by degree of swelling and these apparent rates of reaction which we have measured. And these insights, of course, are interesting, not only because they provide guidelines to allow for the preparation of stable surface-grafted polymer assemblies, but of course, a better understanding of these processes is also interesting if you think about generating force-responsive surface-grafted polymers. Then finally, I would like to thank all the team members for all their contributions over the past 2 decades. These funding agencies for providing the funding for our research, you for listening, and if there is time for questions or comments, I'd be very happy to take them. Thank you very much.
Thank you very much, Professor Klok, for your presentation. We have a couple of questions from the audience, so would be great if you can answer it for us. So at some point you mentioned that there is this correlation between the swelling and the solvent properties. Could you explain a little bit more what kind of solvent properties would you mean here, and whether that's possible to predict what kind of solvent would cause what kind of swelling ratio and then the future degradation?
Yeah, so basically what I mean is basically in polymer science term, basically the better the solvents, so a good solvent solvates a polymer very well as compared to a less good solvent. For a cross-linked polymer network, solvation basically means swelling, right? For a surface-grafted polymer film, or for a surface-confined polymer film, swelling basically means that this film swells in the y direction, right? So basically a dry film of a given thickness, if it takes up solvent, a good solvent will swell in the z direction, and the amount of solvent this polymer will, or this polymer film will be taking up depends on the quality of the solvent, right? So basically on the polymer solvent interaction parameter, if you would want to quantify this. So theoretically, we haven't done this for experimental reasons, right? But if you would want to have let's say a polymer physics type of approach, my idea would be to correlate a Flory-Huggins chi parameter, which is actually a quantitative measure of solvent quality, right? With these reaction rates, quote unquote, which we have been trying to measure.
Okay, thank you very much. So the next question is, what makes the grafting different from this solution? Are they similar processes, or can they be thought about them differently? So you mentioned that the grafting rates correlate more with polymer stretching than dielectric constant. Could you a little bit elaborate on this?
Can you repeat the question again?
So what makes the grafting different from the dissolution of these polymers?
What makes the grafting different from dissolution of the polymers?
So are they?
Yeah. So basically, degrafting and dissolution are 2 different things. So degrafting is basically, if you look at the experiments that we have done, they show basically that the rate at which this happens, or basically how fast degrafting occurs, depends on the swelling ratio, right? So it's not just basically a process that happens spontaneous, but it needs to be triggered by solvent uptake, and it requires these polymer chains to stretch, right? Dissolution would mean also that basically, if you look at this cartoon, if it would just be dissolution, then the end point of this degrafting process would be a bare substrate. But the end point of this degrafting process is not a bare silicon substrate, but it's a silicon substrate covered with a thin polymer layer, because the degrafting process continues. Basically, the degrafting process is accompanied by a continuous collapse of these polymer chains. So basically, this degrafting process transforms polymers that initially have a highly extended conformation into polymer chains that have a collapsed conformation. So as you go from the left to the right on this slide, tension in these macromolecules decreases. And we believe that as a consequence, this also reduces the driving force for this chemical reaction that leads to degrafting. If it would be dissolution, right, that would basically mean that if you wait sufficiently long enough, everything would be washed away from the surface.
Okay.
So this correlation, the correlation we observe between the swelling ratio and the time frame with which it happens, or the rate actually, is I think an important experiment to differentiate, basically, that differentiates what we call degrafting with what you were referring to as dissolution.
So I will combine a few questions. So there's a question about whether you can share your thoughts about the scaling up in this approach, and particularly whether you can see that this work impacting mechanochemists who are interested in driving chemical reactions via using force, not just the bond breaking, but also bond formation.
So basically, from a polymer brush perspective, I think there are two sides to this. One important aspect to this is basically, if you look at polymer brush films, they have attracted quite some interest for applications that include biosensing or biomaterial surface modification. And for these applications, of course, you would want to have a brush film that remains stable during a certain period time of use. And so being able to conceptualize this degrafting process in the way as I've illustrated here also provides a way to avoid degrafting. So it provides a rational approach to prepare robust surfaces that do not undergo this. That's basically the technological impact into this. If this can be used in one way or the other, can you repeat the last part of your question?
So whether it is possible to, well, how do you see this work impacting mechanochemists who are interested in driving chemical reactions via force? So not only bond breaking, as you have here, but if you want to form a covalent bond.
Yeah. I haven't thought about this. It's a good point. Like I said, my way of looking at this is more along the sides as shown here. So basically, I think there are quite some examples of polymer materials that we are using, which are used in the solvent-swollen state, right? So surface-grafted polymer brushes are a nice model system, but not so widely used, right? But macromolecules in solution find applications, but in particular, crosslinked polymer networks are widely used. So I actually, the way I like to look at this is basically, for me, our findings on surface-grafted polymer brushes kind of inspired me or triggered me to think more about solvent-swollen crosslinked polymer networks. And I'm interested if basically swelling of cross-linked polymer networks in good solvents, if these networks would undergo similar type of processes, right? So basically, if you look in cross-linked polymer networks that we use as super absorbers or for biomaterials applications, would similar types of effects, so basically bond breaking reactions, also play a role there? And would this affect network properties in a way that we haven't seen before? This is more the types of problems we have been thinking about.
Okay. And the last question is that in many of these slides, you show that there is a range of forces that you think would be acting of the order of between 10 picoseconds to 100 picoseconds, a little bit more. So you have this degradation rates. Have you ever tried to model what's the magnitude of the forces that are happening on this covalent bond that trigger the degradation? The grafting?
No, we have not done so, but I would be interested in doing that. So we have not thought about modeling. We thought about experiments, right? This is basically the last example I threw in. We have been trying to think about, and we are trying to think about experimental ways to measure those forces, but actually modeling this I have not done yet. But if there is anyone who is interested in joining forces, I would be very happy to discuss. Yeah.
That's a perfect place to advertise your needs. Okay, so this is all questions that we had from the audience. So thank you very much for your presentation. And
very welcome. Thanks a lot. Have a good day.
Okay, so thank you, Professor Klok, for outstanding presentation, and thank you very much for joining us. I would like to remind you that all of our previous seminar speakers are available to watch anytime on our YouTube channel, and I encourage you to check them out. Also, I would like to remind you about the survey. I will be very grateful if you can spend a few minutes to provide us a feedback. And finally, we have some great seminar speakers planned in the upcoming months, and we hope that you will join for all of them. Thank you again.